Good morning, students. Myself, Professor Fularani, working in the Department of Agriculture, GGST College, Haryana, Shahpur. The topic for my today's presentation is sporulation and germination in bacteria. Under this topic, we will cover the types of spores and sporulation and germination stages. What is sporulation? Sporulation refers to the formation of spores from vegetative cells during unfavorable environmental conditions like radiation, extreme heat, cold, lack of nutrition, etc. The Greek meaning of spore is seed. Spore is metabolically dormant structure produced during unfavorable conditions by the process called sporulation. Metabolically dormant means the vegetative cell in the form of spore is not multiplying, but it is live in nature. Sporulation occurs during the late log phase or early stationary phase because of the starvation of food. Under favorable conditions, spores germinate to give vegetative cells. Spores are resistant to nutrition starvation, temperature, extreme pH, antibiotics because it has high dipicolinic acid content, high calcium concentration, and high amount of SASP, small acid soluble DNA binding proteins. About the structure of spore is made up of exosporium, spore coat, cortex and core. In my last presentation I have covered in detail the structure of the spores. You can get reference of PPT. Types of spores. Spores are of four types. Number one, endospores. Endospores are produced within the cell. They are produced many strains of Clostridium species, Bacillus species, and Sporosarcinia species. If we compare the other types of the spores, this endospore is the most defensive structures that is capable of surviving in the adverse conditions. And exospore means outside the cell, and this is the specialty of actinobacteria and methylocyanus species. These spores are formed outside with the help of a process known as the budding, and these cells can survive in adverse environment conditions better than the vegetative cells. The third form of the spores are cysts. Cysts basically are the group of cells when the conditions are unfavorable, these cells are joined together and form a group, and this is the specialty of azotobacter species, and they are less resistant to tough environmental conditions in comparison to the endospores. And fourth category is myxospores. Myxospores is a type of spore that is produced by myxobacteria or myxobacteriaceae family. They are also known as the slimy mold bacteria, and they are formed within the fruiting bodies of the bacteria. Now coming to the stages of sporulation. Sporulation, there are different stages through which the vegetative cell has to pass to reach its final stage, that is the spore. And during unfavorable conditions, vegetative cells convert into spore by the process known as sporulation. Sporulation can be divided into several stages. In Bacillus subtilis, entire process of sporulation takes 8 hours to complete from stage 0 to stage 7. So in this slide you can see the vegetative cell on the left side on the top, that is vegetative cells. It start its sporulation process, and the first stage is the axial filament formation and the division of the chromosomes, and then there is the formation of four spores. After the four spore formation, other stage is the engulfment stage. In the engulfment stage, what will happen, the chromosome of the mother cell will disintegrate, that is not used for the formation of the spores, and the other is from the cortex. And after the cortex there is the formation of the exosporangium, that is the containing the very thick layers of dipicolinic acids, and in the stage of sixth, it is further fifth and sixth stage is further converted into the eighth stage, that is the eighth in the mother cell will release the spores after its disintegrations. Now the spores are can survive in the environment that is an adverse condition, either the temperature is high or very low, or there is no nutrition for the cell, and during this the cell can survive in these conditions. Now we will discuss all these stages one by one. First stage is the zero stage, that is the vegetative cell, and second stage in the first stage there is axial filament formation stage. What will happen, the bacterial chromosome become thread like, that's why it's known as the axial filament, and axial filament attached to the cytoplasmic membrane by the mesosome. Mesosome is again is a infolding of the cytoplasmic membrane, and during this process cell will enlarge, and the beta PHBA, that is the polybeta hydroxybutyric acid in the Bacillus species, it is used as a reserve food, and that is utilized during the sporulation. And in the second stage there is the that is known as the four spore formation. In four spore formation there is asymmetric cell division occur, so that 30% of the chromosome will be one side and 70% will be the other side. What will happen, cell membrane form the septum near one end, which enclose a small portion of DNA forming the four spores. Now these four spores further engulfed, that is process is no third stage is known as the engulfment of the four spore. Who will engulf the four spore, mother cell membrane grow around the four spore and will engulf it, and four spore now has two membrane layers, that is it is now multi-layer. And stage 4 and 5 it's known as the cortex and coat assembly. Synthesis of the peptidoglycan cortex followed by formation of proteinaceous spore coat. In this stage, spore is enclosed in two different layers that include the outer coat consisting of different types of proteins and cortex that consist of a special type of peptidoglycan. And next stage is the sixth stage, that is the lysis of mother cell. The mother cell undergoes lysis or disintegration. Some material from the mother cell may use to build on the spore coat. And the last stage is the spore release. Mature spores are released into the environment, and the and capable of surviving in high temperature, or you can say in the adverse conditions. When the environment conditions are normal or they are favorable, what will happen, these spores can germinate again into a vegetative cell. Now this is a process of germination, that is from the spores conditions, when from the spore conversion to the again to the vegetative cell. Endospores remain dormant for years. Now you can imagine the why these gram-negative bacteria are so pathogenic in nature, because they can produce the spores, and inside the spores these cells, the bacteria can survive for not for 1 or 2 days, for years. For under favorable conditions, each endospore germinate to give rise to a vegetative cell. There are three stages. Suppose conditions are favorable, it will not immediately start germinating, but will happen, it will first endospore will activate in first stage. How it will activate itself, it will activate with the help of activate with the help of some what will say some sort of molecules or factor molecules. These effector molecules will initiate the activation. Once this activation has been started, the spore coat, that is the thick layer, will start damaging itself. It is also known as the autolysis, and in the germination state, one will have the activated spores now start germinating, and with the help of factor molecules, binding of the factor molecules activate autolysis and destroy. What will autolysis will do, it will destroy the peptidoglycan layer of the cortex, peptidoglycan layer of the cortex. And after destruction of the peptidoglycan, what will happen, water will enter, that is used by the cell, and it will swells, and the calcium dipicolinonic acid will released. It is also start disintegrating, and now what will happen in the last stage, that is the outgrowth. In the outgrowth stage, the water swelling of the spore occurs, and along with swelling, synthesis of DNA, RNA, and other proteins will occur. A small germ cell emerge out after breaking the spore coat, and it will begin to grow into a vegetative cell. Means it will start multiplying as it is a normal cell. Thank you.